Our last speaker to wrap up this first segment in the morning is Dr. Sachin Patel. Sachin is the James G. Blakemore Professor of Psychiatry and Behavioral Sciences, Molecular Physiology and Biophysics, and Pharmacology. And he's also the Director of the Division of General Psychiatry at Vanderbilt University Medical Center. Sachin's lab is focused on elucidating the mechanisms by which the brain regulates the stress responses that are relevant to trauma and stress-related psychiatric disorders. His group is specifically interested in the role of endogenous cannabinoids as modulators of stress responsivity, and he has active research projects in the areas of endocannabinoid synaptic biology, preclinical-based therapeutics development, and investigation of circuit-level mechanisms by which endocannabinoids modulate stress-related behavioral phenotypes in rodent models. Through his research, Sachin ultimately hopes to gain novel insights to the pathophysiology of and novel treatment approaches for stress-related neuropsychiatric disorders. Sachin, I too have my small personal anecdote, if you will. When I first met you at Vanderbilt a few years back, you had just invested in our technology, as I recall, and I remember sitting in that conference room with you, fielding one question after another, not only about the methods and the details that many PIs honestly don't sometimes get into, but also about how you'd ultimately be able to translate the data to insights that would help the folks you see in the clinic. Sachin, it's a real honor for us to be in a position to help translational researchers like yourself, like Terry Ressler, who of course who just spoke. And I think the audience today is in for a real treat as you present on the translational implications of endocannabinoid modulation. Sachin, the virtual stage is yours.
Great. Thank you, Kunal and Pushkar, for the kind invitation, and for Inscopix for this amazing summit you're putting on today for everyone. I'm going to go ahead and share my screen. Oh. Hold on one second here. Okay, I hope folks can see this okay. So I'm going to spend the next 10 or 15 minutes talking to you all about our interest in endocannabinoids and how they regulate stress responsivity and the implications for that knowledge for the potential new treatment development for stress-related psychiatric disorders like post-traumatic stress disorder that Carrie just spoke about. So we've heard a lot about this and the year of woe, as Tom has eloquently put it. And just to give a short summary, that we all know stress is a major risk factor for the exacerbation of all forms of mental illness, whether that's psychotic breaks in patients with schizophrenia, relapse to substance use disorders in patients, as well as, of course, major depression and other anxiety disorders. And so understanding the mechanisms that translate stress exposure into affective pathology obviously could have broad implications for then mitigating the effects of stress and improving mental health in times like we have today. And so our focus over the past 10 or 15 years has been on a particular neuromodulatory system known as the endocannabinoids. And as their name implies, they're highly related to exocannabinoids that come from cannabis plants. And it's no secret that there is a strong interaction or a strong relationship between cannabis and stress responsivity. For example, one of the most common reasons people cite for cannabis use is reduction in anxiety, intention, and stress relief. And importantly, stress coping motives and symptom coping motives are highly cited in patients that have PTSD that use cannabis. And this, of course, is well known to industries like the cannabis industry that has been marketing different types of cannabis formulation in places where it's legal to, for example, help cope with stress or to cope with anxiety and tension relief, for example, is shown here with these different types of cannabinoid products. But what we're going to focus on are endogenous cannabinoids. So these are molecules that are made by the brain and other parts of the body that mimic some of the effects of exogenous cannabinoids. And the focus of this talk is going to be on this molecule 2-arachidonyl glycerol. It's a brain lipid that's produced by brain cells and other cells within the body. And it acts at the same molecular target, this cannabinoid receptor, as the active component of cannabis plants, which is THC or tetrahydrocannabinol. So it's the endogenous cannabinoids that are produced by your brain. And we're interested in how these compounds might be important for physiologically regulating stress responses. And this system is highly active within brain regions that we've already heard a lot about, including the amygdala, which is sort of the emotional center for fear, anxiety, as well as positive emotions, and a little bit about a brain area that we haven't heard too much about, which is the prefrontal cortex. And coupling between these areas, or functional connectivity between the amygdala and dorsal parts of the frontal cortex, are strongly associated with the generation of negative affect in humans as well as rodent model systems. And this is just one example of that from our collaborators here at Vanderbilt, showing a functional imaging study under conditions of either neutral or potential threat stimuli. And what you can see is when Dr. Blackford seeded the amygdala and asked what other parts of the brain are connected when people are exposed to potentially threatening stimuli, you see areas of this dorsal prefrontal cortex light up quite nicely. And importantly, the coupling between the amygdala and the cortex is strongly correlated with trait anxiety. And trait anxiety is a predictor for mental health disorders such as major depression. And so we can also look at this from a rodent model perspective and ask, is this a translationally or a evolutionarily conserved neural circuit, which might allow us to study this in reduced models like rodent systems? And we can see here in this particular experiment that when we image prefrontal cortical neurons and expose mice now to brief aversive foot shocks, and you can see the video playing of the types of neural activity we can record, we see many different types of neurons in the prefrontal cortex that are activated, some in response to the shock and some response to the shock offset, and some show reductions in activity. So clearly this area is active in rodents, and more importantly, inputs from the amygdala to the prefrontal cortex are also activated by stress, as you can see here in the purple increase in the calcium signal from these axon terminals. So this is an evolutionarily conserved amygdala-cortical circuit that's stress responsive and important for generating fear and anxiety-like states. And if we can understand the mechanisms by which stress enhances coupling within this circuit, that might give us insights into the pathophysiology of stress-related disorders, as well as potentially reveal new opportunities to intervene and reduce this circuit strengthening and potentially affect symptoms of stress-related disorders. And so in a particular model we looked at was, again, exposure of animals to a brief foot shock. And this particular stimulus results in an anxiety-like states in these animals measured, for example, 24 hours later as an increase in avoidance of this open arm in this an elevated plus maze. And I don't want to get too much into the details, but lower time here represents an increase in anxiety-like responses. And we can then measure connectivity between the basolateral amygdala and the prefrontal cortex using a combination of optogenetics and single-cell electrophysiology. And the main finding from this work by David Marcus, recently published earlier this year, was that there seems to be an increase in presynaptic excitatory glutamate release from the amygdala to the prefrontal cortex after this stress exposure. And we can measure that using a measure known as the paired pulse ratio, but see it visually here as these glutamatergic currents showing a much higher release probability 24 hours after stress exposure. So we know that in this particular model, there is an increase in connectivity between the amygdala and the prefrontal cortex that is associated with an increased anxiety-like state, paralleling what we know about this circuit in humans. And what he found during the course of experiments was that prefrontal cortex neurons will release these endocannabinoids, 2-arachidonoglycerol, that activate receptors located on these presynaptic terminals arising from the amygdala. And when those receptors are active, what they do is reduce the release of neurotransmitters like glutamate. So this is a feedback mechanism aimed at dampening or reducing connectivity between these areas and ultimately reducing then anxiety and stress responses. And what we found was that after stress exposure, that this system becomes highly compromised, such that there's an increase in excitatory neurotransmitter glutamate release from amygdala inputs to the prefrontal cortex. And so stress compromises endocannabinoid signaling in a way that results in increased connectivity between the amygdala and the prefrontal cortex. And so if this breakdown in this cannabinoid feedback system is actually causally related to the generation of anxiety, for example, after stress exposure, if we can go in and specifically disrupt this system artificially, can we recapitulate some of the effects of stress at the synaptic level and the behavioral level? And the way we did one experiment was to selectively delete cannabinoid receptors from these amygdala inputs to the prefrontal cortex. And when we did this, we were actually able to replicate the effects of stress at the synaptic level. So you can see in this particular manipulation where the receptors are missing from the amygdala inputs to the cortex, these synapses are now in a high release probability state, so then increase in connectivity between the amygdala and the prefrontal cortex. And similarly to stress, we can actually see an anxiety-like phenotype emerge in these animals. So now we've actually caused a stress-like state by simply compromising endocannabinoid signaling within this particular neural circuit without the animals ever actually having been exposed to stress. And so the summary of this part of the talk is that stress exposure results in an increase in coupling between the amygdala and the dorsal prefrontal cortex. And we think that increase in coupling is actually caused by a deficiency in this feedback system that's mediated by endocannabinoids. And of course, the next question is really, how can we fix this? If we know what's causing it, how can we use that information and then potentially revert the brain into a low anxiety state? And so one of the things we thought about was, well, if deficient cannabinoid signaling is pro-anxiogenic and enhances coupling within this aversive circuit, what happens if we increase levels of 2-AG? And we can do that, for example, by blocking its degradation and inhibiting this enzyme monoacylglycerol lipase. And when we do that, we can see increases in brain levels of 2-AG, and we can also see a reversal of the anxiety effect caused by stress. In this particular assay, you can see an anxiety-like effect after animals are exposed to stress, and a reversal of that effect when we elevate levels of 2-AG. We also know, as Carrie mentioned, that not everyone that is exposed to stress is susceptible to the adverse consequences or will develop disorders like PTSD. And we can mimic this also in large populations of rodent species. So in this particular experiment, we exposed mice to a stressor and examined their anxiety-like response before and after stress exposure. And if we look here, the increased numbers represent increases in anxiety, and we can clearly see a large group of animals that really show no response to stress, and we term these animals resistant. And there's a different group of animals here shown in red that actually show larger increases in anxiety after stress exposure. And ordinarily, this happens in about 35% of mice that we examine. However, if we treat animals with a compound that elevates 2-AG only before this test period, we can see that that percentage drops dramatically. So not only can we affect the anxiety-like behavior, but we can also potentially modulate the proportion of subjects here showing susceptibility to developing some of these adverse effects of stress. And so, yes, we have some evidence that increasing 2-AG levels could actually revert the neural circuit changes, which I haven't shown you here, but also more importantly, the behavioral effects. And just to point out that there are over 20 patents for compounds that are able to elevate 2-AG levels in the brain. And some of these compounds have undergone early phase clinical trials. So I'm optimistic that within the next 3 to 5 years, we may potentially have novel treatment approaches based on this cannabinoid system for stress-related psychiatric disorders. The last thing I want to talk about for the next 2 or 3 minutes is this concept of fear generalization. So Dr. Ressler pointed out many aspects of PTSD pathophysiology. This is one that we've been interested in for the past few years. And it's related to conditioned fear responses. So when people are exposed to trauma, they form associations between environmental contextual cues and that traumatic experience, such that re-exposure to those cues will then trigger those re-experiencing events, as well as the physiological and emotional sequelae of that trauma. That's normal. We use those processes to avoid danger in the future. However, what's not physiological is the progressive generalization of those cues, such that no longer is it the sound of a gunshot, for example, that would trigger that re-experiencing, but that can generalize to loud noises like a car backfiring, and in the most extreme case, any sort of loud, unpredictable noise. And you can see how this process could lead to avoidance behavior. People don't want to go outside for fear of encountering these cues and then re-experiencing the trauma and feeling those anxiety-like symptoms. So it's a very debilitating symptom domain that's caused by this generalization process. And we can model this in rodents. So in this particular experiment, we paired a foot shock with a particular tone here denoted in red. And then on the next day, when we play that particular tone, the animals will show a fear response. But we can also play a tone that's related but distinct from the conditioned stimulus and ask what happens. And we did this experiment under two different conditions. The first is with intact endocannabinoid signaling, and the second is with depleted 2-AG signaling. And we did this by pharmacologically inhibiting the enzyme that produces 2-AG. And when we did this experiment, we can see that on day 2, control animals show an increase in freezing response or fear behavior when the conditioned stimulus comes on, but not when the novel tone is played. So they're able to discriminate these sensory cues in the environment and mount the appropriate fear response, the freezing or the fear behavior to the conditioned stimulus, but a lack of freezing when the novel tone is played. In contrast, in these animals that have deficient 2-AG signaling, you can clearly see a very large increase now in freezing to the novel tone and a lack of discrimination between the true threat predictive stimulus and a novel sensory stimulus. So these animals are generalizing their fear response. And what we can do then is try and understand what might be happening at a neural coding level by incorporating, for example, single-cell calcium imaging within the prefrontal cortex, which is critical for this type of behavior. And so we recapitulated this behavioral paradigm while recording from prefrontal cortical neurons. And you can see, again, the behavior here where animals that are treated with the vehicle show lower freezing to the novel tone and higher freezing to the conditioned stimulus, whereas under the endocannabinoid deficient state, you see comparable freezing to both the novel tone and the CS+. And when we look at several hundred neurons that we can record across novel tone presentation and conditioned stimulus presentation under vehicle conditions and under endocannabinoid depleted conditions, we see many cells that show increases in activity denoted in red, as well as some that show decreases in activity. But if we look at everything together and we look at the net activity of the prefrontal cortex, we see that during novel tone presentation, there's a much stronger increase in activity in the endocannabinoid deficient mice. Now, this is consistent with an increase in freezing behavior when the novel tone is presented and consistent with what we know about the prefrontal cortex and its requirement for conditioned fear expression. We can also then focus in on the neurons that were highly activated during the tone presentation or the CS+ presentation. And here in my last slide, you can see that under vehicle treatment conditions, that most of the cells that are activated by the novel tone are in fact not activated by the conditioned stimulus and vice versa. And you see this nice double dissociation. And in fact, the majority of cells respond only to one sensory stimulus, either the conditioned stimulus or the novel tone. However, under 2-AG deficient conditions, you lose that double dissociation. You can see that here quite nicely, where you have many cells that are activated during the novel tone and that are also activated during the conditioned stimulus. You lose this nice patterning, if you will, and these cells are not staying in their lane anymore. And quantitatively, you can see that now over 35% of cells are actually responding to both stimuli. And so we think that the ability of these neural sensory inputs to be coded for by different populations is really important for animals to show distinct behavioral responses, freezing to the conditioned stimulus and not freezing to the novel tone. And that when endocannabinoid signaling is deficient, that segregation is lost. And that large overlap in the neural representation of the novel tone and the conditioned stimulus might actually be related to that fear generalization under endocannabinoid deficient states. And the corollary of that is, of course, if we can enhance 2-AG signaling under some circumstances, are we able to reduce fear generalization and really therapeutically attack one of these core symptom domains of PTSD? And so in summary, low levels of 2-AG signaling, we believe, are associated with high levels of anxiety, stress reactivity, increased amygdala-PFC coupling, and fear generalization and impaired extinction, which I haven't showed you here. And we're hopeful that use of compounds like MAG lipase inhibitors that increase levels of 2-AG will be able to mitigate many of these effects and be potentially novel therapeutic approaches for stress-related disorders. And this data also could explain the high comorbidity between cannabis use and people that suffer from anxiety-like disorders. So if you have deficient endocannabinoid signaling, it might be that you're finding that activating the cannabinoid receptor through cannabis use might be putting you into a higher cannabinoid signaling state. And so this might provide a framework for attacking, for example, problematic cannabis use or cannabis use disorder in addition. And so with the general theme of today's summit, I want to charge folks with talking about mental health. And this is an amazing first step in terms of this summit in and of itself. And I've put up here some advice from NAMI about how we can do that. This can be done whether you have $1 or $1,000 to donate. Everyone can begin about talking about mental health today. And it's something that is really going to be required for the fundamental change in our country, the way we view, treat, and approach mental health.
Thank you so much, Sachin. That was a fascinating talk, and as you can imagine, there are a whole host of questions. But in the interest of time, we'll just take a couple from the audience here. The first actually picks up from what you just closed with. Do we know if humans exposed to chronic THC use start producing less endogenous 2-AG? Perhaps then would that lead to withdrawal anxiety-like behavior?
Yeah, that's a great question. So I don't think we actually know about whether chronic cannabis use will reduce production, but what we do know through human PET imaging studies is that chronic cannabis use does actually downregulate the receptor. So it is likely that chronic cannabis use will produce a deficiency state, especially during withdrawal. That definitely is mediated at the level of the receptor. It may also be at the level of the ligand, but we don't have really clinical data on that. But certainly chronic cannabis use will have negative consequences in terms of down-regulating this important physiological system.
Makes sense, makes sense. And the second question here is, have you found that the feedback mechanism between the prefrontal cortex and amygdala synapses is uniform? Or do you think there may be differences based on amygdala afferents in distinct PFC layers, layer 1 versus layer 5, 6, or subregions, PL versus IL?
Yeah, so we've done a lot of those experiments, and I can tell you that this is more specific for superficial layers, so layer 2, 3, and actually is only seen in a reciprocal circuit. So the only cell type that shows this particular synaptic compromise are cells that project back to the amygdala. So it's a closed excitatory loop that seems to be most affected by this endocannabinoid compromise after stress. And we know that those reverse projecting cells are primarily within the superficial layer, so that matches up with our laminar analysis also.
Great, great. Awesome. Well, we could take more questions on this topic. It's a very, very interesting topic. But unfortunately, in the interest of time, we'll call it a wrap here. Sachin, thank you so much.